Welcome to our channel Bio by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, today's topic is fertilization. As we have already learned about oogenesis and spermatogenesis, now we have to learn how fertilization is occurred. So today's topic is fertilization. As during copulation, sperms are delivered into the female reproductive tract through vagina and cervix. So sperm pass through vagina, cervix and fallopian tube, where ovum is already being present in ampullary isthmic junction. Sperm, they move 2 to 3 millimeter per minute with the speed of 2 to 3 millimeters per minute. And this is what happened during intercourse, also called coitus. So at the time of coitus or intercourse, when sperms are delivered, they are rushed to or through reproductive tract and reach to the ovum where fertilization occurs. As I told you that the ovum is present in the ampullary isthmic junction. These are the fimbriae. They help to collect the ovum. When ovum reach to the ampullary isthmic junction, by the time if simultaneously after copulation, sperm also reached to the same portion, that fertilization occur. Otherwise, all the copulations are not necessarily mean that fertilization will occur, because simultaneous movement of sperm and ovum, that ovum to be reached there and sperm also has to be reached there. Ovum may be there for 2 to 3 days, that is when it is secreted, released on 14th day, it may wait 14, 15, 16, 17 days. It is alive by the time if sperms are released and reach to the ovum. So it is necessary to have simultaneous or synchronized reaching of ovum and sperm at ampullary isthmic junction to have the fertilization. Therefore, it is said that all the copulation not necessarily mean to have the fertilization, as if ovum is released but sperm is not released, or if sperm is released and ovum is not released, then there is no fertilization. That is the what meaning of that all the copulation need not to reach to the fertilization. Yes, we have to understand in detail how sperm encounter the ovum and fertilization occur through the process of this fertilization. So to understand fertilization, that is what it can be explained in 3 steps. First step is that contact of sperm and ovum. Second step, activation of egg. And third is fusion of pronuclei. That means nucleus of the male and nucleus of the female. Now, as we already learned during oogenesis, that on 14th day when ovulation occurs, although it is said that it is ovulation, but ovum is not released. What exactly it is released? Secondary oocyte arrested in metaphase stage. Is it clear to you? I hope you had seen my last video of oogenesis, where it is clearly shown and explained that on 14th day, it is, instead of saying ovulation, it can be said as a secondary oocytation. So that secondary oocyte arrested in metaphase which released as it is in the same condition, it get into oviduct through fimbriae, and columnar ciliated epithelium cell help that secondary oocyte arrested in metaphase stage along with zona pellucida and corona radiata to move to the ampullary isthmic junction. So to understand this contact of the sperm and ovum, that sperm secrete anti-fertilizing and ovum secrete fertilizing, that enable to have the contact of. So to mention there how the contact in between these two, that ovum secrete fertilizing and sperm secrete anti-fertilizing. These two components help to combine or meet or sperm and ovum to come in contact. So by this diagram, you can understand that here the sperm secrete fertilizing and secrete anti-fertilizing and ovum secrete fertilizing, enabling them to come together. So to understand, I too can draw diagram and explain you. So here the ovum with zona pellucida around it, also called pre-vitelline space, because this pre-vitelline space is in between, just above the cell membrane, plasma membrane. The zona pellucida is acellular. Zona pellucida is acellular. So this is zona pellucida. This is acellular and it is thick. It is thick. And all around zona pellucida, there is a layer of corona radiata cells. There is a layer of corona radiata cells. See, these are corona radiata cells. So these are the corona radiata cells. 4-5 layers of corona radiata cells. It is not 2-3. So these are the cells of corona radiata which hold by hyaluronic acid. They are held together by hyaluronic acid. Now, as a first step, that contact, that means these cells secrete fertilizing and sperm. And sperm has acrosome, nucleus, middle piece and tail. Now this acrosome secrete this fertilizing. Acrosome secrete this fertilizing, that anti-fertilizing. Acrosome secrete anti-fertilizing. This fertilizing and anti-fertilizing enable them to come in contact, and that is very important to understand. So what is being secreted by ovum? Fertilizing. What is being secreted by sperm? Anti-fertilizing. What is their importance? To make them to come in contact. Now next step is activation of egg. Now when sperm come in contact, that sperm acrosome consists of the sperm acrosome. This structure is sperm acrosome, and all around this has cell membrane with scanty cytoplasm. Now already sperm has proximal centrioles present. These are the proximal centrioles and these are the distal centrioles. The sperm has this mitochondrial sheath having in middle layer, and these are the fibers. They are continue microfibril into the tail also. This is the sperm structure having 23 chromosomes. In that nucleus. Now second step, activation of egg. Under the activation of egg, to learn, under the activation of egg, physical and physiological changes happen. What is physiological and physical changes? That is very important. What is physiological change? Physiological with the entry of Na positive ion, release of Na positive ion here into the acrosome burst and release sperm lysine. Here that egg cell, as soon as that sperm lysine released and break the corona radiata and zona pellucida, in ovum what physiological changes occur? That one, increases consumption of O2 and release of CO2, as well as production of increase in production of ammonia. See, this is very important. Increases production of ammonia. Now see, increase in the consumption of O2, release of CO2 and production of ammonia. That shows that OHM become very active. As soon as this Na positive, very important for NEET, Na positive enter into acrosome, acrosome burst and begin to release hyaluronidase, corona radiata cell digesting enzyme, zona lysine enzyme. See, these are the three components are released from acrosome. Hyaluronidase dissolve hyaluronic acid present here. There is a hyaluronic acid that hold these corona radiata cells together. So what this dissolve, this hyaluronidase dissolve this hyaluronic acid. This corona radiata lysine digests the corona radiata cell and zona lysine make the whole into the zona pellucida as shown in this diagram here. See, this sperm is secreting hyaluronidase to dissolve the hyaluronic acid, corona radiata is disappear. This is a zona pellucida. Now as I said you that acrosome also release zona lysine, therefore this zona lysine digests this zona pellucida. Now after digestion of corona radiata and zona pellucida, now this sperm is about to enter into previtelline space. When it enter into previtelline space, now that tail remain, middle piece and tail remain outside. What exactly enter into the ovum is that sperm nucleus with proximal centriole. Sperm nucleus with proximal centriole and mitochondria and mitochondria. See, as soon as corona radiata and zona pellucida are break, this sperm approaches to the through vitelline space into the plasma of the cytoplasm. Now it develop the reception cone as content of the acrosome are released. That content of the acrosome release enable cytoplasm of this egg cell to form the reception cone. So it form the reception cone as like that when we place a stone into the water, it get just bubbled or form the cone like structure like this, that has cytoplasm, it just get around the sperm nucleus and form the reception cone. That reception cone is this. This is a reception cone. This is a reception cone. So now nucleus with that reception cone is ready to enter. That is called penetration path. So this is a penetration path enabling that nucleus along with proximal centriole and mitochondria to enter into the cytoplasm. Is very important. What is the function of that proximal centriole along with nucleus? When there will be a fertilization and when zygote form, to form the spindle fiber of the first mitotic division of the zygote. So spindle fibers are formed by proximal centriole. I hope you can remember that centriole are made up of, very important, they are made up of 9 triplets so as to form the spindle fiber, and mitochondria provide the energy. Okay. Now as a part of fertilization, now nucleus, proximal centriole and mitochondria are enter into the ovum or secondary oocyte. As soon as this penetration had begun, there the meiotic division, secondary oocyte arrested in metaphase, continue to divide and form second polar body and oocyte. See, as soon as sperm penetrate through corona radiata and zona pellucida, that secondary oocyte arrested in metaphase stage continue to divide and form the second polar body and oocyte, so as to that oocyte is a pronucleus of the female fuse with pronucleus of the male to form the zygote. Wait, to understand it. Now we have just completed an activation of egg. This is a physiological change. Then what are the physical changes? It's very important. That is what I explain you. These are the physical changes which I did explain to you. Physical changes. That is to form the penetration path, copulatory path and penetration path. Penetration path that I told you degenerate the corona radiata and zona pellucida and form the reception cone. Together it is called penetration path, and that is the physical changes. So physical changes: 1. Degeneration of or eroding of. Degeneration or eroding of corona radiata cells and zona pellucida. Second, that cytoplasm of the ovum. So this is what secondary oocyte arrested in metaphase stage. Now, as a physical changes, that corona radiata and zona pellucida is degenerated, and nucleus along with secondary or proximal centriole and mitochondria enter into, and by forming reception cone. So second point is formation of reception cone. So formation of reception cone. So this reception cone form at the animal pole by the time that second meiosis arrested in metaphase stage is completed and form the oocyte and ovum. To know it, we can have reference of this diagram. Here they have not shown that how secondary oocyte undergo mitosis arrested in metaphase stage and form. To understand that, I am going to draw a diagram to enable you to understand how that two pronuclei get fused. That is very important. So formation of reception cone. So I am showing here a reception cone by the time that second meiotic division is also completed. So here there is a reception cone. Here this cytoplasm is slightly elevated above. So cytoplasm is elevated above. No doubt, all around there is a corona radiata. This is what zona pellucida. This is zona pellucida. It's continue around this. Second meiosis is completed, and oocyte is porous. This is a oocyte, and this is a second polar body. Second polar body supposed to get outside through the animal pole. Nucleus, male nucleus, enter along with proximal nuclei and mitochondria. Now this continue move in 180 degrees called copulatory path. So penetration path is completed. Now copulatory path. This granulose, cortical granules, as shown in this diagram, will be very easy to understand you. See here, cortical granule word is shown. These cortical granule later they form a cortical granules. They form after fertilization at the same place here. After fertilization, they form here. This will be closed. Cortical granule will secrete the content. Will form very thick layer called fertilization membrane to stop the entry of another sperm and prevent the polyspermy. This is also one of the event that is called physical event, but formation of fertilization. So third is after fertilization, formation of fertilization membrane. Fertilization membrane to prevent the polyspermy. Now here these two pronuclei are ready to fuse and form the zygote. By the time corona radiata is already degenerated, this corona radiata is already degenerated. So degeneration of corona radiata is completed. Fertilization membrane is formed. Here there will be formation of fertilization membrane to prevent the polyspermy, because many sperm that they approach, these are the suppose sperm. many sperm that they are very close vicinity, but these sperms are not allowed to enter since that fertilization is completed and fertilization membrane is formed. So formation of this fertilization membrane prevent fertilization membrane prevent the polyspermy. It prevent, very important to understand, prevent polyspermy. This polyspermy is common in birds, not in mammal. So here this pronuclei, fusion of these two pronuclei completed and form the zygote. So fusion of these two pronuclei. So once again, to understand that fertilization here, suppose here the fertilization is completed, second polar body, this released out second polar body, that released out, this is a zygote, and zona pellucida is still continue. Zona pellucida is still continue all around it. So zona pellucida is continue, and this is what zygote is suppose ready to have the continuity of the division. So corona reducta get degenerated when sperm penetrate, but zona pellucida will continue up to blastula stage of the embryonic development. Here the fertilization is completed. We did understand that how when ovum release and reach to the ampullar ischemic junction, by the time when sperm release, fertilization occur. The process of fertilization I did explain you through this diagram, and by this explanation, and you could have understood how this physical and physiological changes enabling this sperm to enter into the secondary oocyte. Then by the time this oocyte which is produced is ready to fertilize, this is called oocyte, is very important to understand. This is now real ovum which form after penetration of sperm, and when that penetration of nucleus along with proximal centriole and mitochondria. Again, please remember this one is very important to understand. Here these mitochondria, these are the proximal centriole and sperm nucleus. This sperm nucleus, these three together, this is a sperm nucleus. So these three together penetrate into, and that is called penetration path. Now they move through 180 degree cytoplasm of the ovum, 180 degree, that is called copulatory path. By the time this second polar body emerge out and fertilization membrane is formed, pronuclei combine and form the zygote, and thus the fertilization is completed. So with this diagram you get clarification that where the fertilization occur, how the sperm penetrate so as to deliver the nucleus along with proximal centriole and mitochondria to form the zygote. By the time that ovum arrested in metaphase stage, continue to divide form oocyte and second. So second polar body come out through animal pole, and that animal pole, which we call this one, get closed by the fertilization membrane formed by the secretion of cortical granules, and this fertilization membrane is thicker than zona pellucida to prevent the polyspermy. And through this copulatory pathway, when this nucleus completed movement 180 degree combined with female pronuclei, that is oocyte, male nucleus and female nucleus combine to form a zygote with 2n number of chromosomes, and that is what total number of 46 chromosome, and thus it is a zygote ready to divide and redivide to form these 2 cell stage, 4 cell stage, 8 cell stage, morula stage, and in blastula stage they get implanted for the further embryonic development. I hope you did understand this entire process of fertilization, what I did explain here. Even if you are in doubt, you can ask in comment section. And yes, as it is clearly mentioned, there are many NEET questions, MCQs can be asked, and we shall discuss as a separately in the separate part of our video which MCQs can be asked in NEET examination. So with this concept, you are in position to answer all the MCQs which supposed to be asked, and therefore conceptual study is very important to understand the question and apply your knowledge to solve the MCQs in NEET examination. As a part of board's examination, what is fertilization? Explaining, you can comfortably explain with this information. By the way, that diagram I have been drawn stepwise, that is sufficient to have the understanding and explanation in board examination. I appeal you to subscribe and like our channel, as well as press bell icon so as to receive new videos as and when they are uploaded. Thank you.